Hi, good evening. This is Khadija Bobo, and today we'll be talking about somitogenesis. And the outline will be just a preview into what is a paraxial mesoderm. We're going to see segmentation, that is the formation of somites from somites and somitomes from pre-somite structures. And then we're going to see the differentiation of somites after the formation of somatocele space and somatocele cells, and which it would differentiate into the ventromedial sclerotome and also dorsolateral demimyotome. And also then we have a clinical application of what we're going to discuss today. So basically we'll be discussing about classification of a mesoderm. You remember we talked about we have the after the formation of the three germ layers, we have the paraxial mesoderm. That is the mesoderm that is found just lateral to the neural plate and notochord. You could see it out in red. Then you have the intermediate mesoderm, which differentiates into the urogenital structures. And then you have the lateral plate mesoderm, which splits into two, with most of it split forming the sclerotome and somatopia and splanchnopluric structures. So you can see it in the diagram below. The yellow is indicating the paraxial mesoderm. The tiny red is indicating the intermediate mesoderm, and then you can see the two structures. The purple one is going to give rise to your somatoplure, and also the red, the blue on the bottom is going to give you the splanchnopluric. Then this green structure is below the developing spinal cord. You can see the spinal tube there, and then ventral to it is the green structure, which is the notochord. So this is just telling us that this is the posterior view of a developing embryo, and you can see that the superior and the cranial and caudal part of it are still open. This is called the neural pore, the ventral and the rostral and caudal neural pore. It means that the fusion of the neural plate has started occurring from the midline and then goes cranially and caudally. So lateral to it you find elevations there, and that is the formation of the somites. So this somite structure, which in this diagram is shown in red, is basically the proliferation and differentiation of the paraxial mesoderm as the neural plate continues to fuse in the midline. So this is a section of it so that we can see the interior aspect of the somites. You can see the somites on the lateral aspect of the diagram, and then on the superior aspect, after that's the cross section, you can see part of it going to yellow, some of them are in red, and then the most lateral aspect of it. Don't forget, in purple there is the neural tube. Just beside the purple is the neural crest cells, which are very important because they stimulate the structures in that area to differentiate further. Then you have the notochord, and ventral to the notochord is the developing dorsal aorta. So remember what we had at the end of the third week, we have the neural plate gradually forming the neural groove. You can see it in blue on top there, and then which gradually fuse together, and you can see the mesoderm layer, which is in red, is gradually differentiating. That is in diagram C to give rise to the different aspects. You can see that the somite with a middle space, that middle space is called the somitocele, and it's being formed by arrangement of cells which separates the somatoceles from the cell, and those are called the somatocele cells, which we'll see later on. You can see the small intermediate mesoderm, and then you can see the lateral plate mesoderm, which has split into the somite, the somatic and the splanchnic mesoderm. The yellow in that diagram is indicating your endoderm, or the the roof aspect of the yolk sac. And then gradually in D, you can see how the neural tube has formed, and then ventral to it, you can see a small round structure, which is the notochord. And D, you can see two dorsal aortas, which in E will fuse together to give rise to one structure. You can see the body cavity also that has fully formed in E, while in D, it's still connected to the outside amniotic sac. All right, so that is basically what we're talking about. And then somitogenesis is a differentiation or proliferation of the paraxial mesoderm, which in this aspect is going to differentiate back to epithelial type of cell. You remember it was an epithelial type of cell from the epiplast, then became the mesoderm, but this is going to differentiate. But I don't want to disturb you on that molecular aspect. For those BSs that want to study further, they will also specialize in that aspect. So somite starts to differentiate. It starts to aggregate by the 19th day and the 20th day. You tend to see the first somite form, and it is called somitomeres. That is why those ones found in the superior aspect of occipital regions. Each somites are then just separated by what is referred to as the intersomite spaces. And after that, you have the formation of 3 pairs of somites every day. And then at the end, you're going to have a total formation of 7 somitomeres. And somitomeres are ones found within the skull. Then we have 4 occipital somites. And then definitely one would degenerate. We end up having 3 that would differentiate to give rise to an occipital bone. And then you have 7 cervical, and some textbooks refer to it as 8 cervical. And we have 12 thoracic, 5 lumbar, 5 sacral. Then we have 8 to 10 coccygeal forming. So by the end of the 5th week, we have about 42 to 44 pairs of somites formed. So these somites are going to differentiate into 3 different structures. It's going to differentiate into the dermis of the embryo, is going to give rise to the myotome of the embryo, is also give rise to the sclerotome of the embryo. Note that the epidermis of the embryo or the human body is going to develop from the epiblast, while the dermis and the fat subcutaneous tissues develop from the dermatome, while the myotome give rise to the muscles of the body and the body wall alongside the limbs as well. The sclerotome give rise to the axial skeleton, and we're going to see how those things differentiate. So you can see the arrows showing you the movement of structures around the blue neural tube and also around the notochord. We're going to see that. So the fate of each somite is going to give rise to an axial skeleton. You can see the axial skeleton includes the skull, the vertebral bones, and the ribs as well. And also don't forget the sacrum and coccygeal. So it also gave rise to the dura meninges as well as the dura associating dura blood vessels. It gives rise to the intrinsic back muscles, and as well as the body wall muscles, which include the muscles of the limbs as well. And then give rise to the dermis of the skin, which includes the dermis and the hypodermis fat collection. So first of all, you have in this diagram, the first diagram is just a representation of a 2-day chick embryo, and then you have a 3-day embryo to 4-day embryo, which has a similar differentiation pattern with that of the human being. So first, what you do is you have the formation of the somatocele, and that's the space around. You can see in the first diagram, you can see an a round space inside the somite, so that is the somatocele, and then there is a layer of cells that are arranged on that inner circular structure, which is referred to as the somatocele cells or atromere cells, which give rise to the cells of the von Ebner fissures. In the future we're going to see that. So you can see that the blue diagram is representing the sclerotome, while the orange diagram is representing the demimyotome. So the sclerotome is going to divide itself into two major portions, and the one that is very close on the medial aspect and also the ventral aspect will give rise to the sclerotome. That is called the ventromedial portion of it. And then you can see the ventrolateral, the dorsolateral, it's in the posterior aspect, and on the lateral aspect gave rise to the dorsolateral demimyotome. Then this cells tends to migrate. So in the second diagram you can see the blue cells dissociating and also trying to go around the neural tube as well as the notochord in the midline. So the third diagram is showing more of the cells of the sclerotome going round in that aspect. And then you can also look at the demimyotome has segregated into two layers, and you can see the cells migrate and they dissociate and migrate. So you can see the myotome are the ones found on the more lateral aspects and more ventral aspect as compared to the dermatomes. So in this diagram, it's just a similar diagram from the previous diagram, but here you can see at C, from the diagram C, you can see more of the cells of the sclerotome going ventral aspects. You can see SLCA and SLCL, so that is called the sclerotome anterior aspect, and you have the sclerotome the lateral aspect. You also have the sclerotome posterior aspect, and also you have the sclerotome medial aspect, which give rise to the different parts of the structure. We're going to see the different fates they are going to give rise to. Don't forget the DM is our demimyotome, but in the diagram D you can gradually see it separating into two. You can see the lateral portions of it giving rise to the myotome, while the dorsal aspect of it giving rise to the dermis. So this is just a nomenclature of the diagram you see, and then you get to find the differentiation. So you can see in this diagram A and B, you can see how the blue structures, which is the sclerotome, goes round the neural tube, and as well you can see the notochord, which is in A as a black structure just below the neural tube. And also you can see the migration of the red structures in B going around the body as well. That is the somatic mesoderm is going into the somatic layer of the mesoderm. This is the migration of the myotome, which you're going to see in detail as well. Alright, so on the most posterior aspect, you see the migration of that. So in a closer detail, this is what is happening. So you can see the migration as A and B in this diagram. So like we've explained, you remember that during the folding of the neural tube, you have the neural crest give off. So the neural crest will stimulate alongside the notochord, also stimulate the paraxial mesoderm to differentiate, to undergo somitogenesis. So now you can see the cells are in different colors. You can see the sclerotome in blue, and then you can see an orange. In dark orange you see the myotome, and in light orange you could also see the dermatome. Alright, so in between the myotome and the sclerotome there is a purple color of cells called the syndertomes. The syndertomes are very important because they give rise to the particular part of the transverse process, as well as we're going to see later in the future. So here you can see how those cells are gradually migrating. You can see the sclerotome in purple going to surround the notochord as well as the a neural tube, and then you can also see the red myotome also dissociating and going round as well. And then you also see the dermatomes, which give rise to dermis, also dissociating and lining on just below the ectoderm. So in this diagram is showing you that as well. So you can see the neural crystals have also stimulated for the formation of the dorsal ganglia, and the dorsal ganglia are important because the formation of those dorsal ganglia are going to be located in the midline mostly of each of the sclerotome, each of the somites. And it's important because it gives rise to a particular interesting landmark. So in this we said that this sclerotome give rise to the vertebral bones. So we're going to start talking about the ones that encirculate the neural tube and notochord, immediately giving rise to the vertebral column. So first you have the formation of the notochord. We've discussed about that, and a notochord is very important. And then you have the formation of the mesoderm layer. You remember when we had that. We also have the important formation of the sclerotome, which goes to surround the notochord and neural tube. And then afterwards, they resegment. There's a resegmentation, and then the rearrangement of the structure, which we're going to see. And mesenchymal cells undergo chondrification before ossification, which are important. In the next class, we're going to talk about ossification of bones. So in this diagram we have in the first stage, you can see the neural tube in blue. It doesn't have the spinous processes. And then you can see the sclerotome also, and then laterally you can see the red myotome. Alright, so this first one, the one on the left more, you can see it's been labeled. You have the neural tube, and then a particular point in the middle of the where you have the location of the sclerotome, you have bulging. That bulging is important because that's the location where you had the formation of those posterior ganglia, and then they relate to the formation. You have the routing of nerves in that aspect. And don't forget, at that level you also have the somite. The somite will be separated into two. You have a higher dense area and a lower less dense area. It means the higher level above that dotted line you have more cells, and then below that dotted lines you have less cells. So you can see in 1, 2, 3 somites, and then each of those portion, the higher portion and the lower portion, are being separated by these dotted lines, which is the von Eppner fissure. And those von Eppner fissure are made of those somatocele cells, if you remember. So what happened is that those cells tend to move to separate those two portions, and when they separate these two portions, it gives us allowance space that allows for these growing nerves to go and meet the muscle. So you can see the second blue structure with the diagrams beside it. So now you can see they have interspinal nerves that goes at that von Eppner space to go and meet the myotome at each level. So you can see that the upper the cranial dense structure, somite, part of the somite, and then you can see the lower less than part of the somite, and then still above the spinal nerve you can see another upper aspect of it and lower aspect. So that was how they separated. So what happened is that the lower part and the lower part of the segmental somite above will fuse with the upper part of the following somite. That is what happens throughout. So in the third diagram you could see that the vertebral bones have formed. Each of these vertebral bones is formed from the lower aspect of the upper somite and the upper aspect of the lower somite. So in this diagram where you have pink, yellow, pink, yellow, pink, yellow, so the pink in this diagram in the first somite here, which is referred to as O4, occipital 4 somite. Alright, so occipital 4 somite here is showing that the upper part is pink and the lower part is yellow. The upper dense part and the lower less dense part. Then you can see that a C1 has a pink, it has the upper dense part, a pink lower dense part. C3 has an upper pink dense part. Sorry, C2 has an upper pink dense part and a lower less dense yellow part. So you can see that this the foramen magnum, alright, is made of the lower less part of O4 and the upper dense part of C1. Then C1 is made of the lower less dense part of C1 and the upper high dense part of C2, and that is how the story continues. So you end up having the cranial nerves 1, 2, 3 till 8 moving on top of the vertebral preceding it. So this diagram is also trying to explain this structure we've seen. So now here you can see the myotome on the lateral aspect. Now we're seeing what is the cross section of A. So in the cross section of A you can see the myotome. Then you can also see in the midline you can see the notochord. Alright, so in this diagram we don't get to see the cross sections, we don't get to see the neural tube. Okay, so what we see is that you see the sclerotome, and then in between every sclerotome is an intersegmental arteries. So this intersegmental arteries is also moving alongside the intersegmental nerves we've seen in the previous diagram. So what happened is that we need, you can also see the nerves, the nerves are growing where you have the space, where you have an imaginary line. Remember the von Ebner fissure? So here you can see each sclerotome is made of a higher gray and a lower black area, all of them. So what happened is that these somites, the first somite here, this first sclerotome, you get to see the gray and black. So what happened is that the nerve is growing at that junction where the gray and black meet. That is where you have the von Ebner line, and then you also have the growth of the nerves in that area. So what happened is that they split into two. So this lower black will come and join the second sclerotome gray. So in the diagram B you can see what is happening. So now every vertebral bone, the black is on top while the gray is below. The black is on top while the gray is below, as it is vice versa with what was in the sclerotome. And then another important thing is that you can see where you have the notochord, the black notochord there. Where you have the vertebral bones, the notochord is disappearing. It only persists where you have the vertebral discs. Okay, where you have the vertebral discs. So what happened is that the notochord that is found in that area, it gives rise to the nucleus pulsi of the vertebral discs, while the cells of the von Ebner, or this sclerosyl cells, give rise to the annulus fibrosus of the intervertebral discs. So in this diagram we're still explaining what we've seen earlier on. So you remember, you can see in A, you can see the blood vessels in between the somite, each of the sclerotome. Okay, so each of the sclerotome you get to see an intersegmental artery there. All right, then you can see a nerve trying to go and meet the myotome at the position of the bone ulna. You can see the nerve leaving the neural tube to go into the muscles or the myotome on the lateral aspects. Okay, so what happened is that the dark yellow aspect is the cranial sclerotome, which is more dense, while the lower aspect is less dense, okay, and it's light brown. And then you also see orange. Then you see the upper part is dense and the lower part is light. All right, so what happened is that at those bone ulnar lines they move away. So what happened is that the caudal sclerotome of the superior sclerotome fuses with the upper part of the lower sclerotome. So if you look at B, that is what is happening. So now where you have the space, it's not at where you have the arteries, but where you have the nerves. So if you compare A and B, you see the difference. All right, so now you've seen the positions have changed. So if we assume the first sclerotome here is sclerotome 1 and 2, 3, what happened is that in the B diagram, the lower portion of 1 will face the upper portion of 2. The lower portion of 2 will fuse with the upper portion of 3, and that is how it continues. All right, so what happened is that the new notochord also in that position in the body of the vertebra, it disappears except where you find the discs, so it remains as the nucleus pulsi of the intervertebral discs. All right, so this is what we're talking about. So can you see that? So this is the cranial dense portions. All right, and then they will become the caudal portions of the vertebral bones. All right, you can see they become the caudal portions of the vertebral bones. So this is also explaining the same scenario. You can see the first diagram is just talking about the migration of the structures from the somites, and then the ones that migrate around the notochord. You can see the notochord in the middle. It is sclerotome. The next diagram is also showing you the same thing. And then if you come to this upper diagram on the lateral where it's been cycled with the big purple is intersegmental artery, and then you can also see the vertebra. So you can see the shading of the structures as well, and then gradually they change their positions. So the notochord, only the notochord found in the intervertebral discs persist as the nuclear pulsi of the intervertebral discs. This is also another diagram trying to describe that resegmentation. So you can see in blue, in diagram A, you can see blue is telling you that is the neural tube, yellow is the notochord, and then what you see on the lateral aspect is the somites, and then gradually the somites differentiate into different categories, and then you have the sclerotome going ventrally and dorsally to encirculate the notochord and also the neural tube. So now in the diagram below, you can also see how the nerves are forming from the neural tube as well, and then you can see how those have encircled the notochord, and then you can see the blue and orange section, and there's a space between them. Gradually you can see the blue section is just telling you that is where you find the presence of the notochord and notochord. So you can see everywhere in the diagram 6, you can see all the notochord have disappeared except that small area which is referred to as the nucleus pulsi. When you come to diagram B, it is just basically telling us where the location, the differentiation, the part of differentiation, the blue, and the blue is going to form the body, and then you have the cranial processes, and you also have the lamina, and purple, you have the transverse processes in purple, and also you have the ribs also forming as well. And all the structures are important because different parts of the sclerotome give rise to different parts of the vertebral bones, as we're going to see. So still explaining that, I think this diagram will show more light. Is you can see this as somites, and then you can see somite 4, 5, 6, and 7. So you can basically get to appreciate the differentiation of that. So you can see 4 and 5 and 6 in blue. So the green is telling you that's the green area, the pink and the red is telling you that is that area, the blue is telling you that is that area. So what happened is that the lower aspect of this green, the lower aspect, after resegmentation, you're going to have a change in position. So this lower aspect is going to fuse with the upper aspect of the next one, and then you have the lower aspect of the red is going to fuse with the upper aspect of the 6, and then the lower aspect of the 6 is going to fuse with the upper aspect of the 7, and that is why you have the darker colors above instead of in the resegmentation it is more above than below, so they've changed their position. So the formation of the vertebral bones, just like we've talked about, you have migration, and then they've moved and differentiate. And don't forget that they also form the they form the intervertebral disc, except the nuclear pulsi of the vertebral disc. So part of the sclerotome, we have the ventral part of the sclerotome, which becomes the body and the disc of the intervertebral disc. Then the dorsal part of the sclerotome forms the spinous process and the lamina. Then we have the lateral part or syndytomes, form the distal ribs, tendon of the intrinsic muscles of the back. Then you have the medial or the meningeotome part, forms the meninges and the blood associating blood vessels of the spinal cord. Then you have the central part, which forms the articular pillar of the pedicels and the transverse processes. So here we're still going to explain about the resegmentation of that structure. So you can see in this diagram we have somites on the lateral aspect. You can see NT is the neural tube, NC as the notochord, and diagram A. So you can see the yellow and blue. Okay, so blue is blue on top, yellow below. That is what it continues. So you can see the position of the somites before resegmentation. All right, so basically blue is on top, yellow is below, blue is on top, yellow is on below, and in the diagram. All right, so so in the somites, yellow is on top, blue is below. All right, so R is rostral, C is caudal in the somite. So the somite is telling us that R is rostral, C is caudal. So the superior, yellow is up, blue is down. That is what it means. So now look at the direction of movement. All right, So basically what you're going to end up with, you can see where you have the von Ebner line. All right, and then gradually what happens is that resegmentation happens. So where you see resegmentation, it means that it's going to change its position. And if you look at B, blue becomes on top and yellow becomes below. Blue becomes on top and yellow becomes below. That is what happened. And then when we talked about the different parts of Somite, you can see what we talk about different parts of Somite. The maroon is telling us is the ventral aspect of the Somite. Green is telling us is the lateral part of the somites, and then you can see the purple there is telling us as the dorsal part of not somites, sclerotome. So maroon, ventral part of sclerotome, green, lateral part of sclerotome, orange, central part of sclerotome, and then you have the dorsal part of sclerotome. All right, so basically when you come to this diagram, in the bottom you can see with the feet of it. So the ventral becomes the body and the discs. Then you have the green, which is the lateral. You can see it just a little bit of the pedicels and the transverse process, and then you can see the orange is going to come to form the central, which is going to form the lamina, and the rest. Then you have the dorsal, which is going to form the transverse processes. All right, and so here is also telling us the different aspects of it that is forming the different structure. So in A, you can see it's just the middle, the cells in the middle, in the SM, all right, which is the somites, and then the middle you find the somitocils, and then the ventral aspect you see the red, which is called the sclerotome, and then the green is demimyotome. And then you can see how the sclerotome differentiates into the ventral aspect, central aspect, lateral, and dorsal aspect. When you come to C, you can see how gradually the ventral aspect is going to form the body. And then you can see the central forming the lamina as well, and then you can see the dorsal in blue forming the transverse processes and spinous processes, the spinous processes, while the L is forming just a little bit of the ribs, as well as little bit of the transverse process. All right, so, and this is also just another diagram to describe that. So this diagram is also talking about the same thing. You can also see the dorsal aspect. You can see the meningeal aspect of it that forms the meninges. You can see the ventral aspect in green. In then you can see the lateral aspect of it, and then behind you can see the sclerotome, the demimyotome. You can see the myotome, and then you can see the syndactome cell separating the demimyotome from the sclerotome aspect. So here you could also see how they go and surround the neural tube and the notochord as well. And then here you can find the final structure that has been formed. As this structure forms, they undergo ossification, and then they have two ossification centers. They have the primary ossification center and the secondary ossification centers. In the next class, we're going to talk about bone ossification, that is bone formation, the two types of bone formation, and then we'll talk about the primary and secondary ossification centers, and then I believe you'll be able to understand ossification centers more. And clinical application of this is there's several things that could happen if and the neural tube does not fuse on the posterior aspect. You might have a side effect, and then you could also have and in a case where the neural tube has formed, the meninges have formed, but the lamina and the spinous processes has not formed. So you have the different type of meningocele conditions and the spinal bifid. Next is the differentiation of myotome. So the myotome have two different parts. The dorsomedial part of the myotome and also the ventromedial part of the myotome. So dorsomedial part of the myotome give rise to intrinsic muscles of the back, the myspelon of the muscle, and also the extensor muscles of the limbs, while the ventral lateral part of the myotome give rise to the muscles of the body wall and flexor muscles of the limb, while the dermomyotome give rise to the dermis and the blade of the scapula, as well as the subcutaneous tissues of the body. So we can say that the myotome have, we could also see the myotome and the occipital myotome give rise to the muscles of the tongue and some of the muscles of the head and neck. And as in association with the pharyngeal arches, will give rise also to the muscles of the head and neck. And then you can see the different stages, and you remember when we get to stage C, you can see the remaining structure is the dermomyotome, and then you can see that the muscles also differentiate into two groups. You have the epimeres and the hypomeres. So the epimeres are the dorsomedial, and then the hypomeres are the ventrolateral myotomes. So the epimeres, you remember, are going to give rise to the intrinsic muscles of the back and the extensor muscles of the limbs, while the hypomeres give rise to the hyperaxial muscles, which include the rectus, abdominal oblique muscles, intrinsic and extrinsic abdominal muscles, as well as the thoracic, external, internal, and innermost layer of the thoracic muscles, as well as the flexor muscles of the limbs. Thank you very much. If you have a question, don't forget to drop your questions on the WhatsApp group or the Slack. Have a nice day.